Hello, friends, everybody, good day. Today, I would like to discuss about the C4 rice gene editing. What is the C4 rice gene editing? So, it seems that everybody uh, knows that rice is a C3 plant. So, how to convert it is a big project. So it is an ambitious project aimed in improving the efficiency of rice plants photosynthesis by converting them from C3 to C4 photosynthesis system through gene editing. So initially the originally rice is a C3 plant. So it should be converted to gene modification by gene editing, not modification, I'm sorry, gene editing to C4. So what is it that we get the benefits of converting a C4 pathway? This could dramatically increase the rice yields, particularly in hot and dry climate. Rice being a C3 plant uses a less efficient photosynthetic pathway than C4 plants like maize or sugarcane, especially under conditions of high temperature, intense sunlight, and drought. So that's why to improve the photosynthesis of rice in C3 rice, so gene editing is done to improve the yields, and it will be especially under conditions high temperature, intense sunlight, and drought, and less water needs, and efficient water needs, and the efficient nutrition. It will take less than C3. That's why we need a gene editing for the future farming to meet our population demand for food. So this is what today I would like to discuss with you. So next is this is the sea rice gene editing. This is the vision, the visuals that I am having, which you can see in this. So what is the C4 rice project and gene editing? C4 project and gene editing, the C4 rice project aims to transfer more efficient C4 photosynthesis pathway to rice using gene editing technology like CRISPR. That is one technology is by using this CRISPR technology. By introducing the gene responsible for C4 photosynthesis into rice, the goal is to increase yield, reduce water and nutrient use, and make rice more resilient to climate change. You know that climate is changing very tremendously, very because of our because of our activities, the climate is changing. So we should find out resilience to climate also. This gene editing technologies like CRISPR Cas9 allow precise alternation to be made in the plant's genome. In the case of C4 rice, gene editing is used to introduce or modify the genes necessary to develop a C4 pathway involving complex regularity networks and anatomical changes in leaf structure to support the photosynthesis system. So the process involves identifying and editing gene that control leaf anatomy and enzymes that drive the concentration of CO2 in certain cells, regulatory elements that activate these processes only under specific environment condition. This is what the C4 RISE project and gene editing means. So in total, so gene editing and what is the process involved, we have understood now. Now the next slide is, this is C4 photosynthesis versus C3. What is C4 photosynthesis? What is C3 photosynthesis? C3 plants means most plants, including rice, using the C3 photosynthetic pathway. In this pathway, C3 photosynthetic pathway, the enzyme Rubico fixes CO2 into 3 carbon compounds, hence 3,3, that is why called C3. 3 carbon compounds fixing the called C3. However, Rubisco can also bind to oxygen, leading to photorespiration, which wastes energy and reduce productivity, especially under stress conditions. That means stress condition means highlight, heat and drought. So you know that this will inefficiently, C3 will be very inefficient in also bind to leading to oxygen photorespiration, which wastes energy wasted. In C3, while it is in photosynthesis, there will be so much energy waste. So whereas in C4 it will not be wasted. In C4 plants like maize and sugarcane use an additional step to concentrate CO2 in specific cells, making photosynthesis more efficient by reducing photorespiration. Whatever we lose in C3, here it will reduce the losses of photorespiration. This process allows C4 plants to thrive in hot, sunny and dry environment and produce more biomass compound compared to C3 plants under the same conditions. So when there is high light, heat and drought, C4 plants act more efficiently by reducing photorespiration so that it gives the biomass compared to C3 plants under the C4 conditions. This is what the difference between C3 and C4. So next is, what is difference between GMO and gene editing? So here is what I have explained in the slide. G4, C4 RISE project and gene editing. C4 RISE project aims to transfer the more efficient C4 photosynthesis pathway to rice using gene editing technologies like CRISPR by introducing the gene responsible for C4 photosynthesis into rice. The goal is to increase yield, reduce water and nutrient use, and make rice more resilient to climate change. What is gene editing? So gene editing technologies like CRISPR Cas2 allows these things. This is what I showed just now. I am telling. So the next is, what is the here is some disturbance. Just wait. This is the difference between the GMO and gene editing. So, what is the GMO? Genetically modified organism and gene editing. There is a difference between the GMO, genetically modified organisms, and gene editing. Here, the things are two different gene modifications or can be done GMO through and gene editing. But in the C4 rice, only gene editing is done to improve the yields under certain bad climatic conditions. What is the GMO refers to? The organisms that have been modified by inserting gene material, and that is DNA from a different species into their genome to introduce new traits. This typically involves traditional genetic engineering techniques where foreign genes are introduced to confirm traits like pest resistance, herbicides tolerance, and improve nutrition. Process is in GMOs a gene from another organism, that is from bacteria, whether bacteria, virus, or any other plant animal species, is inserted in the target plant's genome using techniques like agrobacterium, mediated transformation, and gene gum methods. For example, Bt corn is a GMO, genetically modified, where a gene from a bacterium, Bacillus thuringiensis, is inserted into corn to make it resistant to pests. What is the regulation? GMO face stringent regulatory process because of concerns over potential ecological impacts, food safety, and ethical issues. The introduction of foreign DNA can trigger concern about gene flow into wild species, allergically and unintended side effects. So example, Bt cotton resistance to pests, golden rice fortified with vitamin A, Roundup ready crops, herbicide resistance. So this is what about genetically modified organism. The gene editing is definition. What is the gene editing definition I am giving? So that is gene editing is especially with technologies like CRISPR Cas9 involves making precision changes or deletions in an organism existing genome without necessarily introducing foreign DNA. So there will be no introduction in this gene editing foreign DNA. It allows scientists to alter specific genes within a species to either enhance desirable traits or knock out undesirable ones. Gene editing involves, say, within the specific genes within the species to either enhance desirable traits or knock out the undesirables. The process is like this. Gene editing tools like CRISPR enable targeted modifications to a specific gene within the organisms. For example, CRISPR can be used to knock out a gene that makes plants susceptible to disease or to edit a gene to enhance drought tolerance. Unlike GMOs, gene editing can involve only small changes to the organism's own DNA or involve transforming gene within the same species for closely related species. What is the regulation? Since gene editing can involve more specific changes, sometimes only a few pairs of genome. Some countries have fewer regulatory hurdles for gene-edited crops like GMOs, uh, crops than GMOs. This is because of gene editing mimics natural mutation that could occur spontaneously in nature or through traditional breeding. See, example, disease resistant wheat, where a gene making is it's susceptible, it's susceptible to mildew, is edited out. Non-browning mushrooms, via CRISPR technology it is done, and improving drought tolerance in crops by editing gene that regulate water usage. So there is a difference between genetically modified organisms, but genetically modified organisms have some limitations where maybe some see social society may not accept, but gene editing it is not so that there is no limitations, but within the species of the gene only, gene in the species they modify. They won't. There will be no introduction of other genes of species into another species. This is what exactly mean difference between GMO and gene editing. So future of C4 rice and gene editing. C4 rice, the successful conversion of rice from a C3 to C4 plant could significantly improve food security, particularly in regions with high temperature where rice yields are constrained. C4 rice would use water and nitrogen more efficiently, potentially offering high yields and greater climate resilience. So here is gene editing again as a CRISPR. What is CRISPR? The full form is I told CRISPR technology by using gene editing is done. So that is full form is clustered regularly interspaced short palindromic repeats, that is clustered regularly interspaced short palindromic repeats, and other gene editing technologies continue to evolve. They are likely to become a major tool for developing crop varieties tailored to specific environments and challenges including disease resistance, drought tolerance, and improved nutrition content. Sustainability. GMOs and gene editing can make agriculture more sustainable by reducing the need for chemical inputs like pesticides and fertilizers, and by enhancing the resilience of crops to changing climates. The C4 rice project aims to transform rice into more efficient plant by editing its gene to the use of C4 photosynthesis pathway, which is more efficient in hot and dry environment. Gene editing like CRISPR technologies is a precise tool that modifies the organism's own genes, often making small changes, while GMO technologies involves inserting foreign genes into an organism genome. Both approaches hold immense potential to address food security and agriculture sustainability, but they differ in terms of process, regulation, and public perception. But in public perception, gene editing is more acceptable. Gene, GMO technology involves so many constraints That means, so it is a long process, and it is also is not legally, see, in any country, it is not accepted totally by the society of that country. So this is what I would like to tell today. What is a C4 rice? How it is useful for the future food, meat to the growing population. This is one of the most recent technology where we can understand the things, how we can meet the food problem of the growing population of the global world. This is what I mean. Thank you. So thank you very much. This is what I would like to tell you. So this is what I told you. So if anybody has any remarks on this, they can send the email also, or you can send it through I see their questions by seeing my video. And recently I am giving a gap of more days to release my videos on the future farming, because so it takes everybody a little bit confusion of technologies, and it is not updated today's technology. It is only a future technologies. So that is why I am giving very um, lately the next technologies, and but I will give you totally, and after finishing each technology of future technologies, I will give you total in one stroke also, that where you can say. Why this is I am giving in English means most of the people outside of the state also, they can understand what is future technology coming. That's why I am giving. Thank you very much. This is what I